Today, we're talking about the discovery of cells so that we can define life via cell theory. And our success criteria is being able to describe the three main principles of that cell theory. So we know now that all organisms are made up of smaller units called cells, and that cells are the functional unit of life. They're little chambers within our bodies that act as their own units. And we were able to discover these cells because of the microscope and allowed us to see microorganisms, but also see that similar to microorganisms, living things were made up of smaller units too. There wasn't just smaller life that was invisible, there were smaller parts of visible life too. So cells were first discovered by a guy named Robert Hooke. And Hooke lived in the 17th century, and he looked at pieces of cork under a microscope. And cork is made of the bark of the cork tree. And what he saw is that there's these little tiny chambers within the cork. And he thought those chambers looked like the little rooms that monks lived in, so he called them cells, because that's what the rooms were also called in monasteries. What he didn't know was that these cells were actually the dead parts of a living tree. He knew that the cork came from a tree, but he didn't realize the significance that those cells were actually incomplete. They were living things originally. And he included images of those cells within his book of diagrams of objects from underneath microscopes. Anthony van Leeuwenhoek, a Dutch merchant, was the first person who actually described single-celled microorganisms. Uh, he also observed many different types of cells as well. And he called these single-celled organisms animalcules. He thought that they were just really, really tiny, tiny animals, which, as a first assumption, isn't a bad one. We know that many of these microorganisms are, in fact, other forms of life. We wouldn't classify them as animals today, but he was basically trying to describe them in the best terms that he could. He had no way of knowing that they were genetically distinct from what we call animals. So he published that information and hoped scientists could use that information to try and more thoroughly explain the basis of life. And Chalandine and Swan ended up actually doing that. They put together Hooke's information and Leeuwenhoek's information and developed something that we call cell theory. Both of them thought that the cells were actually crystallization of non-living matter, so abiogenesis. And we know now through Virchow's experiments that no, it is actually biogenesis. Cells have to come from other cells. But the key component that they contributed was not the biogenesis or abiogenesis, but that all living organisms were made of one or more cells. What they realized is that many of Leeuwenhoek's animalcules were actually made of a couple cells or even sometimes single-celled organisms. And it was the exact same thing that Hooke was seeing in his cork. So, from cell theory, we get three main principles. We get the idea that all organisms are made of one or more cells. In fact, most organisms that we see are made of billions and billions and billions and billions of cells. The cell is the smallest functional living thing. So there are things smaller than cells inside of them, organelles and DNA and all that good stuff that we will talk about in a little bit. But those are not alive. On their own, they're not a living thing. You can take DNA or a protein out of a cell and it's not alive. Being alive is something that the cell has to do with all of its parts together. And finally, cells can only be produced from other cells. Now, there are major differences between cells. So a plant cell looks different from an animal cell, looks different from a bacterial cell. But all types of life that we know of on planet Earth follow this cell theory. They're made of cells, those cells can produce more cells, and the cell itself is the smallest thing that can actually be considered independently alive.